Have you ever wondered what you would see if you could journey inward, past the surface of your skin, past the intricate layers of your muscles, and down into the very smallest corners of your being? If we were to shrink ourselves down until a single grain of sand appeared as large as a mountain, what would we find at the heart of our existence? We would find that we are not a single solid entity, but rather a vast and bustling civilization. We are a collection of trillions of tiny, vibrant, and incredibly purposeful units. We are, at our most fundamental level, a world of cells. In this first step of our journey together, we are going to explore this invisible universe. We are going to uncover the principles that govern every living thing on this planet, from the oldest redwood tree to the smallest garden ant, and of course to you, as you lie here in the quiet of the night. This is the study of cell biology. It is the foundation of everything we know about health, growth, and the continuity of life itself. Tonight, we will not merely recite facts. We will listen to the story of how life organizes itself. We will discuss the great laws known as the cell theory, and we will meet the two primary types of cells that inhabit our world: the simple, ancient prokaryote, and the complex, sophisticated eukaryote. As the world outside grows dim and your thoughts begin to settle, let us turn our attention to the tiny miracles that make your every breath possible. Lesson 1. Introduction to cell biology. To begin our understanding, we must first look at the rules that define life at its smallest scale. In the vast history of science, there have been few realizations as profound as the cell theory. This theory is the cornerstone upon which all of modern biology is built, and it consists of three essential pillars that describe the nature of existence. The first pillar of this theory tells us that all living organisms are composed of one or more cells. Think about the sheer diversity of life for a moment. Picture a massive blue whale gliding through the cold depths of the ocean, and then picture the microscopic yeast that makes bread rise in a warm kitchen. Despite their differences in size and habitat, they share this one fundamental truth. They are both built from the same basic units. You might imagine the cell as a living brick. Just as a magnificent cathedral is constructed from thousands of individual stones, every living being is constructed from cells, where some organisms, like bacteria, are content to be a single, solitary cell, a complete life within one tiny boundary. Others, like us, are multicellular, a grand and complex society where trillions of cells work in concert to create a single, unified person. The second pillar of the theory is that the cell is the basic structural and functional unit of life. This means that the cell is the smallest level of organization that can truly be considered alive. If you were to take a cell apart and look at its individual molecules, its proteins, its fats, its sugars, you would find marvelous things, but none of them would be alive on their own. Life is the result of the way these parts are organized within the boundary of the cell. It is the cell that takes in energy, the cell that grows, and the cell that responds to the world around it. And it is the smallest flame of life that can burn independently. The third pillar is perhaps the most poetic. All cells arise from pre-existing cells. This tells us that life does not simply appear out of nowhere. It is a continuous, unbroken thread. Every cell in your body right now can trace its lineage back through time, dividing from one cell into two over and over again. This chain of life stretches back billions of years to the very first cells that ever appeared on Earth. You are a living testament to that ancient and successful history. As we look at the world of cells, we see that they generally follow two different blueprints, or styles of living. We call these prokaryotes and eukaryotes. The prokaryotic cell is the elder of the two. These are the simplest forms of life, primarily consisting of bacteria and archaea. If you were to visualize a prokaryotic cell, you might imagine a small, efficient studio apartment. It is a single room where everything happens in the same open space. There are no internal walls to separate the kitchen from the bedroom. The genetic material, the DNA that contains the instructions for life, is not locked away in a separate room. It simply floats in a specific region of the cell. These cells are small, hardy, and incredibly successful, having inhabited every corner of the earth for billions of years. The eukaryotic cell, however, is the type of cell that makes up your body, as well as the bodies of plants, animals, and fungi. If the prokaryote is a studio apartment, the eukaryote is a sprawling multi-room mansion. The word eukaryote actually means true nucleus, and that is its defining feature. In this cell, where the DNA is protected within its own dedicated room, the nucleus. But the complexity doesn't stop there. The eukaryotic cell is filled with many other specialized rooms, which we call organelles. Each organelle has a specific job to do. One might be responsible for generating power, another for building proteins, and another for processing waste. By separating these tasks into different compartments, the eukaryotic cell can become much larger and perform far more complex functions than its prokaryotic cousins. Take a moment to let that idea find its place in your mind. The simple, open studio of the bacterium and the complex, organized mansion of your own cells. Both are masterpieces of nature, but they represent two very different ways of solving the problem of how to stay alive. Don't worry about memorizing the names yet. Just try to visualize the difference between the open space and the structured rooms. If your mind is still following along, let us take a moment to look back at how we discovered this hidden world. For most of human history, we were completely unaware that cells existed. We looked at the world with our own eyes and saw only the large and the obvious. The discovery of the cell required a revolution in how we see, and that revolution began in the 17th century with the invention of the first primitive microscopes. In the year 1665, an English scientist named Robert Hooke was looking at a very thin slice of cork under a microscope he had built himself. Cork is actually the dead bark of an oak tree. As he peered through the lens, Hooke saw that the cork was not a solid substance, but was instead composed of thousands of tiny, empty, box-like structures. To him, they looked like the small, sparse rooms called Cellular, where monks lived in a monastery. And so he called them cells. Hooke was looking at the dead remnants of plant life, the hollow walls that were left behind, but he gave us the name that defines biology to this day. Shortly after Hooke's discovery, a Dutch lens grinder named Antonie van Leeuwenhoek took the study of the invisible world to a much higher level. Leeuwenhoek was a man of extraordinary patience and skill. He created tiny, powerful lenses that allowed him to see things no human had ever seen before. When he looked at a drop of pond water, he discovered a teeming, swirling world of living creatures. He saw tiny organisms swimming and spinning, and he called them animalcules, or little animals. He was the first person to see living cells, and including bacteria and even the cells in his own blood. It took nearly 2 centuries after these early observations for scientists to realize that these cells were not just curiosities, but were the very fabric of all life. In the 1830s, 2 German scientists, Matthias Schleiden, who studied plants, and Theodor Schwann, who studied animals, realized that despite their many differences, all the tissues they examined were made of cells. They sat down together and realized that the little boxes Hooke had seen in cork were the same basic units found in a blade of grass or the muscle of a frog. This was the birth of the cell theory as we know it today. There is a fascinating question we must ask ourselves as we explore this. Why are cells so small? Why aren't we made of just 1 or 2 giant cells the size of a hand? And the answer lies in a very important principle called the surface area to volume ratio. As a cell grows larger, its volume, the space inside, increases much faster than its surface area, the membrane that surrounds it. Imagine a small cube. It has a certain amount of skin to let in food and let out waste for the space inside. If you double the size of that cube, the space inside becomes 8 times larger, but the skin only becomes 4 times larger. Eventually, if a cell gets too big, it simply doesn't have enough surface area to support all the activity happening inside. It would starve or be poisoned by its own waste. By staying small, cells ensure that they have plenty of surface to interact with their environment. This is why, rather than being one giant cell, you are a collection of trillions of tiny ones. It is a matter of mathematical necessity and survival. Try to visualize this as you rest. Miles and miles of microscopic skin, all working to keep the tiny interiors of your cells perfectly nourished. It is a vast, efficient network that operates without a sound. So you see, this isn't just a theory in a book or a collection of historical anecdotes. The study of cell biology is the principle that explains the very reality of your health and your place in nature. When we talk about medicine or nutrition or the way you heal after a scratch, we are ultimately talking about the behavior of your cells. Every time you eat a meal, you are providing the raw materials that your cells need to maintain their mansions. When you breathe, you are delivering the oxygen that every single one of those trillions of units requires to stay alive. And most importantly, when you sleep, as you are doing now, your cells are in a state of intense and purposeful activity. This is the time when they focus on repair. They use the quiet hours of the night to divide, to rebuild the cytoskeleton, and to ensure that the genetic library in the nucleus is protected and ready for the morning. Our understanding of the difference between prokaryotes and eukaryotes is also the reason we have been able to conquer many diseases. Because bacteria are prokaryotic studios, and your cells are eukaryotic mansions, scientists have been able to develop medicines, like antibiotics, that specifically attack the studio without harming the mansion. We target the parts of the bacterium that your cells don't have, allowing us to clear an infection while keeping you safe. Furthermore, the cell theory provides us with a profound sense of connection. When you look at the world around you, you are looking at a giant interconnected family of cells. We are all made of the same basic units, governed by the same ancient laws, and descended from the same original ancestors. Whether it is the moss on a stone or the person sitting next to you, we are all part of the same biological tapestry. This is a good place to rest your thoughts for a moment before we continue. You are a masterpiece of cellular organization, a living city that never sleeps. We have now established our foundation. We have explored the meaning of the cell theory and understood the two grand designs of the cellular world. We have walked through the history of those first brave scientists who peered into the dark and found the light of life's smallest units. But as we have seen, every cell, whether it is a simple prokaryote or a complex eukaryote, must have a boundary. It must have a way to separate itself from the rest of the universe. It must have a skin that is not just a wall, but a discerning gatekeeper. Having now grasped the structure and theory of the cell, we are perfectly prepared to appreciate its function. So when we return, we will take our next step together and examine the remarkable boundary that defines the cell. Our next lesson will explore the plasma membrane. We will see how this thin, fluid barrier decides what is allowed to enter the sanctuary of the cell and what must be kept out. For now, let the logic of the cell rest in your mind. You are well constructed, you are part of an ancient lineage, and you are at peace. Lesson 2. The plasma membrane, structure and transport. We have already established that the cell is the fundamental unit of life, the living mansion of the biological world. But as we continue our journey, we must pause at the threshold. Before we can explore the grand libraries of the nucleus or the busy factories of the ribosomes, we must understand the wall that holds it all together. This is not a cold, brick and mortar wall, but a vibrant, intelligent, and incredibly thin boundary known as the plasma membrane. Tonight, we take the next step in our learning journey and explore this gatekeeper. It is one of the most remarkable structures in nature. It is only about 7 to 10 nanometers thick, so thin that you could stack 10,000 of them and still not reach the thickness of a single sheet of paper. Yet, this delicate film is the only thing standing between the organized harmony of life and the chaos of the outside world. We will look at how it is built from dual-natured molecules, how it behaves like a fluid sea, and how it intelligently decides what is allowed to pass through its gates. This is the study of the boundary where the cell begins and ends. To understand the plasma membrane, we must first meet the molecule that makes it possible, the phospholipid. This molecule is a study in contradictions. It has two very different parts that behave in opposite ways. There is a circular head and two spindly tails. The head is made of a phosphate group, and it is hydrophilic, which literally means water-loving. It seeks out water, wanting to be bathed in it. The tails, however, are made of fatty acids, and they are hydrophobic, or water-fearing. They turn away from water, tucking themselves inward to stay dry. Because cells exist in a watery environment, surrounded by fluid outside and filled with fluid inside, these phospholipids naturally arrange themselves into a bilayer, or a double layer. The water-loving heads face outward on both sides, making contact with the water. The water-fearing tails face each other in the middle, creating a protected, oily core. Think of this bilayer not as a solid wall, but as a film of oil floating on water. It is a stable structure, yet it is incredibly flexible. This oily core is what makes the membrane such an effective barrier. Most substances that are dissolved in water, like salts or sugars, cannot easily pass through this oily center. This is the first layer of the cell's security system. However, a membrane of pure oil would be too simple. It wouldn't be able to do the work of a living cell. This is why we use the fluid mosaic model to describe it. Picture a vast, calm sea of these phospholipids. Floating in this sea, like colorful icebergs or boats, are a variety of proteins. These proteins are the mosaic part of the model. Some stay on the surface, while others span the entire width of the membrane. And because the membrane is fluid, these protein boats are not anchored in one spot. They drift. They can slide slowly through the lipid sea, moving to where they are needed. This fluidity is essential. If the membrane were too rigid, it would shatter like glass. If it were too fluid, it would lose its shape. To maintain this perfect balance, animal cells use a molecule called cholesterol. These little molecules are tucked between the phospholipid tails, acting like a buffer. When it gets cold, they keep the tails from packing too tightly and freezing. When it gets warm, they help hold the tails together so the membrane doesn't fall apart. The most important job of this fluid mosaic is selective permeability. This is a beautiful term for a simple concept. The membrane is picky. It is a discerning gatekeeper. Small, uncharged molecules like oxygen or carbon dioxide can slip right through the oily gaps between the phospholipids without any help. They are like ghosts walking through walls. But larger molecules, or those with an electric charge, like ions, find the oily core impassable. For these molecules, the cell must provide a doorway. This brings us to the proteins. Some of the proteins floating in the membrane are transport proteins. They act as specialized tunnels or revolving doors. A specific protein might only allow water molecules through, while another might only allow glucose. In this way, the cell controls its internal environment with exquisite precision, ensuring that the right molecules are in the right place at the right time. As we dive deeper into how things move across this boundary, we see two distinct philosophies of transport. The first is passive transport. You might think of this as the cell simply letting nature take its course. It requires no energy from the cell. It relies on the natural random movement of molecules, which always tend to spread out from where they are crowded to where they are scarce. This process is called diffusion. Imagine you are standing at the back of a quiet, darkened room, and someone opens a bottle of perfume at the front. Slowly, the scent molecules bounce off one another, spreading through the air until they reach you. They are moving down their concentration gradient. In the cell, oxygen diffuses into the cell, and carbon dioxide diffuses out in exactly this way. When the cell provides a protein tunnel to help a molecule diffuse, we call it facilitated diffusion. It is still passive. The molecule is just being given a dedicated path. A very special type of passive transport is the movement of water, which we call osmosis. Water is the medium of life, and the cell must be very careful about its water balance. If a cell is placed in a solution that is too salty, water will leave the cell to try and balance the concentration outside, and causing the cell to shrivel like a raisin. If the solution is too watery or pure, water will rush into the cell, causing it to swell and potentially burst like an overfilled balloon. This is why your body works so hard to keep the fluids around your cells at a constant saltiness. It is a delicate, silent tug of war happening across every membrane in your body. But sometimes, a cell cannot afford to be passive. Sometimes, it needs to move molecules against the natural flow, like pumping water uphill. This is active transport, and it requires the cell to spend its hard-earned energy currency, ATP. A classic and vital example of this is the sodium-potassium pump. This is a protein machine that lives in the membranes of your nerve and muscle cells. Its job is to constantly pump sodium ions out of the cell and pull potassium ions in, even though there is already plenty of sodium outside and potassium inside. Think of this pump as a person constantly bailing water out of a boat to keep it afloat. This pump uses about a third of all the energy your body consumes while you are at rest. Why? Because by creating this imbalance of ions, the cell creates an electrical charge across its membrane, a tiny biological battery. This battery is what allows your nerves to fire and your muscles to contract. Every thought you have tonight is powered by these tiny, tireless pumps working in the dark. Finally, there are times when a cell needs to move something far too large for a protein tunnel, like a large piece of food or a whole bacterium. For this, the cell uses bulk transport. In a process called endocytosis, the plasma membrane actually reaches out, wraps itself around the object, and pinches off to form a little bubble or vesicle inside the cell. It swallows the object. The reverse process, exocytosis, is how the cell secretes materials. A vesicle from the inside fuses with the plasma membrane, opens up, and spills its contents into the outside world. This is how your brain cells release the chemicals that allow you to feel calm and ready for sleep. If your mind is still following along, you might begin to see that the plasma membrane is much more than a bag holding the cell together. It is the interface between you and the universe. Consider the sensation of a soft blanket against your skin. That feeling is only possible because the membranes of your sensory neurons are equipped with specialized proteins that can sense pressure. When the blanket presses against them, these proteins change shape, allowing ions to flow through the membrane and creating the electrical signal that your brain interprets as soft and warm. The plasma membrane is also the site of many medical mysteries and solutions. Many viruses, including the flu or the common cold, have evolved keys that trick the membrane's receptors into thinking they are friendly guests. The virus binds to a receptor, the membrane opens the gate through endocytosis, and the intruder is ushered inside. Conversely, many of the most important medicines we use work by interacting with the membrane. Beta blockers for heart health, for instance, work by sitting in the membrane's receptors and blocking the stress signals from reaching the cell. So you see, this thin, oily film is not just a boundary. It is a sophisticated communication hub. It is a sensor, a filter, and a power station all in one. It is the reason you can maintain a constant internal environment even as the world around you changes. It is the guardian of your cellular home. We have crossed the threshold. We have studied the gatekeeper and understood the complex dance of molecules moving across the border. We have seen how the cell maintains its electrical charge and how it swallows the things it needs to survive. But now that we have passed through the gates, we find ourselves in the vast interior of the mansion. What fills the space between the membrane and the center? It is not an empty void, but a rich, structured environment filled with support beams and moving parts. Let the logic of the membrane rest in your mind. The fluid sea, the floating proteins, the quiet movement of water and ions. It is a peaceful, orderly system. When we return, we will move deeper into the interior. Our next lesson will explore the cytoplasm and cytoskeleton. We will see how the cell maintains its shape and how it moves things from the membrane to the heart of the cell. We will look at the internal scaffolding that gives the cell its strength. This is a good place to rest. Our journey is well underway, and the path ahead is clear. For now, let your thoughts drift. The gatekeeper is on duty, and all is well within. Lesson 3. The cytoplasm and cytoskeleton. We have passed through the elegant selective gates of the plasma membrane, and now we find ourselves truly within the interior of the living cell. If you were to imagine the cell as a grand, bustling theater, the membrane was the front door, and now we are standing on the stage itself. But as we look around this microscopic world, we must ask, what fills the space between the walls? What prevents the ceiling from collapsing? How do the actors and the stagehands move from one side to the other without getting lost in the dark? In this lesson, we are going to explore the internal environment of the cell, the cytoplasm, and the invisible, intricate web of support, known as the cytoskeleton. It is a common misconception to think of the inside of a cell as a simple bag of water. In reality, it is a highly organized, dense, and dynamic workspace. We will learn about the viscous jelly that hosts the chemistry of life and the 3 distinct types of protein fibers that act as the cell's bones, muscles, and highways. As we sit together in the quiet of this moment, I want you to visualize this interior not as a chaotic soup, but as a meticulously engineered masterpiece of structural integrity and fluid motion. As we begin our exploration of the cell's interior, the first thing we encounter is the cytoplasm. This is a broad term that describes everything nestled between the plasma membrane and the central nucleus. Within this region, we find a substance called the cytosol. It is very important to understand that while the terms are often used interchangeably, they represent different layers of the same reality, and the cytosol is the semi-fluid, jelly-like substance itself, the medium of the cell. If you were to imagine a bowl of fruit gelatin, the fruit pieces would be the organelles, and the gelatin itself would be the cytosol. Together, the gelatin and the fruit constitute the cytoplasm. The cytosol is largely composed of water, but it is a thick water, crowded with dissolved proteins, salts, sugars, and the various building blocks of life. It is within this nutrient-rich soup that the cell's metabolism truly hums along. Many of the chemical reactions that keep you alive, the breaking down of sugar for a quick burst of energy, for instance, happen right here, floating in this golden, viscous fluid. It is a bustling environment, yet it possesses a certain quiet order. However, if a cell were only filled with cytosol, it would be a fragile, shapeless blob. It would have no way to hold its organelles in place, and no way to resist the pressures of the world outside. This is why the cell possesses a cytoskeleton. Despite its name, the cytoskeleton is not a rigid, static frame like the bones in your own body. Instead, it is a dynamic, ever-changing network of protein fibers that stretch from the center of the cell all the way to the inner surface of the membrane. Take a moment to let that idea find its place. Imagine a vast invisible web of cables and beams that can be built, torn down, and moved in a matter of seconds. This is the secret to the cell's strength and its grace. The cytoskeleton is made of 3 primary types of fibers, each with a different thickness and a different purpose. The first and largest of these are the microtubules. Think of these as the heavy-duty girders of the cell. They are hollow rods made of a protein called tubulin. Because they are stiff, they resist compression, acting like the structural beams of a skyscraper that prevent the building from being crushed by its own weight. But microtubules are more than just beams. They are also the railroad tracks of the cell. Special motor proteins walk along these tracks, carrying heavy cargo, like vesicles filled with hormones, from one part of the cell to another. The second type of fiber is the microfilament, sometimes called actin filaments. These are the thinnest of the 3. If microtubules are the beams, microfilaments are the flexible high-tension wires. They are made of a protein called actin, and they form a dense, 3-dimensional network just inside the plasma membrane. This network gives the cell its outer shape and helps it withstand pulling forces. Most importantly, microfilaments are the fibers of movement. When a cell needs to change its shape, or when a muscle cell contracts, it is the microfilaments that are doing the work, sliding past one another to create motion. The third and final type of fiber is the intermediate filament. As the name suggests, they are middle-sized, larger than microfilaments, but smaller than microtubules. These are the most permanent fixtures of the cytoskeleton. While the other 2 types are constantly being rebuilt, intermediate filaments are sturdy, rope-like cables designed for durability. They are the anchors. They wrap around the nucleus to hold it in place, and weave through the cytoplasm to ensure the cell maintains its overall integrity even under great stress. And if your mind is still following along, try to visualize the sheer coordination required for this system to function. It is not enough to have tracks. You must have the engines to move along them. This is where we encounter the motor proteins. These are perhaps the most enchanting residents of the cellular world. Using the energy from ATP, the currency we discussed in our previous lesson, these tiny molecules actually have feet that step along the microtubules. One type, called kinesin, generally carries cargo outward toward the edge of the cell, while another, called dynein, carries materials back toward the center. It is a silent microscopic logistics network, moving the mail and the supplies of the cell with tireless precision while you sleep. Without these motor proteins and the microtubule tracks they tread upon, and the cell would be a stagnant pond rather than a vibrant city. We also see the microtubule system at work in the structures that allow cells to move through their environment, cilia and flagella. A flagellum is like a long whip-like tail. Think of the way a single sperm cell swims toward its destination. Cilia, on the other hand, are shorter and appear in vast numbers, like tiny oars on a galley ship. In your own body, the cells lining your windpipe are covered in millions of cilia. They beat in a coordinated rhythmic wave to sweep dust and mucus up and away from your lungs. This entire mechanical motion is powered by microtubules sliding past one another, a masterpiece of biological engineering that happens without you ever having to think about it. It is also worth noting the history of how we came to understand this internal scaffolding. For a long time, because the fibers of the cytoskeleton are so incredibly thin, They were invisible to traditional light microscopes. Scientists thought the cytoplasm was just a clear fluid. It wasn't until the advent of the electron microscope in the mid-20th century that we could finally see the intricate cobweb-like architecture of the cell. This discovery revolutionized biology. We realized that the cell wasn't just a container for chemicals, but a highly structured machine that could exert force and maintain its own geometry. Consider the centrosome, a region usually located near the nucleus. In animal cells, the centrosome acts as the microtubule organizing center. It is the hub from which the microtubule tracts grow outward. Within the centrosome are two small structures called centrioles, which look like tiny bundles of sticks arranged in a circle. While their exact role is still a subject of much study, we know they are essential when a cell prepares to divide. They help organize the spindle that pulls the genetic material apart, ensuring that each new daughter cell receives exactly what it needs to survive. Take a moment to let that idea find its place. Even as you rest, your cells are maintaining these structures, checking the tracks, and ensuring the anchors are holding fast. This is a good place to rest your thoughts for a moment before we continue. So, why is this internal architecture so vital to your everyday life? You can feel the work of the cytoskeleton in every movement you make. Every time you blink, every time you reach for a glass of water, and even the steady rhythm of your heart is the result of actin microfilaments and their partner proteins working in unison. In the medical world, our understanding of the cytoskeleton is a powerful tool in the fight against disease. Consider the way we treat cancer. Many chemotherapy drugs work by specifically targeting the microtubules. Because cancer cells divide much faster than normal cells, they are constantly building and tearing down microtubule spindles to move their DNA. Drugs like Taxol come in and freeze the microtubules, preventing them from being dismantled. If the railroad tracks can't be moved, the cell cannot finish its division and eventually dies. It is a stark example of how knowing the architecture of the cell allows us to intervene when life's processes go awry. Furthermore, we see the importance of the cytoskeleton in the way your immune system protects you. When a white blood cell detects a bacterium in your bloodstream, it doesn't just float there. It must hunt. It does this by rapidly growing its actin microfilaments in one direction, pushing the plasma membrane forward in a crawl. The cell literally reshapes itself to chase down the intruder. It is the cytoskeleton acting as both the muscle and the steering wheel. Even the clarity of your thoughts depends on this system. Your brain cells, or neurons, have long, thin extensions called axons that can reach over a foot in length. To keep those axons healthy, the cell must ship nutrients and the signals from the main body all the way to the tip. This long-distance transport is handled entirely by motor proteins, walking along microtubule tracks. If those tracks break down, the neuron fails, which is exactly what we see in neurodegenerative conditions like Alzheimer's disease. Don't worry about memorizing the names of the fibers. Just let the logic of it wash over you. You are held together by a beautiful invisible geometry. You are strong because your cells are reinforced. You are capable of movement because your cells can reshape themselves. We have explored the environment of the cell and the scaffolding that gives it form. We have seen the jelly of the cytosol, the heavy girders of the microtubules, the tension wires of the microfilaments, and the sturdy anchors of the intermediate filaments. We have watched the motor proteins tread their silent paths, keeping the cellular city supplied and functional. But every city needs a city hall, and every mansion needs a master library where the original plans and laws are kept safe from the chaos of the outside world. Now that we understand the structure of the house, we are ready to meet its inhabitant. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the study of the nucleus and genetic control. We will move into the very heart of the cell to see where the blueprints are kept and how the brain of the cell issues the commands that govern everything we have discussed so far. Our journey continues inward. When we return, we will unlock the vault. But for now, let your mind rest. Visualize the steady, quiet work of the cytoskeleton holding you in its gentle embrace. You are well supported. You are at peace. Lesson 4. The nucleus and genetic control. We have traveled through the selective gates of the membrane and navigated the intricate supporting web of the cytoskeleton. Now, we arrive at the very heart of the eukaryotic cell. In the center of this microscopic metropolis lies a structure that serves as the governing body, the master library, and the architect's vault all in one. This is the nucleus. In this lesson, we will explore the nucleus not merely as a physical object, but as the seat of genetic control. This is where the cell's most precious cargo, its DNA, is stored, protected, and interpreted. It is the nucleus that decides which proteins will be built, how the cell will grow, and when it is time to divide. As we explore this central chamber together, I want you to imagine it as the quiet, hushed room at the center of a great university, where ancient vital scrolls are kept under lock and key, yet their instructions are constantly being copied and sent out to guide the work of the entire world outside. We will examine the double-layered security of the nuclear envelope, the mysterious density of the nucleolus, and the elegant way that miles of genetic instructions are packed into a space smaller than a grain of dust. To understand the nucleus, we must first look at its exterior. Just as the cell itself is defined by a membrane, the nucleus is shielded by its own specialized boundary called the nuclear envelope. However, this is no ordinary wall. It is a double membrane, meaning it consists of two distinct lipid bilayers separated by a narrow space. This extra layer of protection highlights just how vital the contents of the nucleus are to the survival of the organism. If the plasma membrane is the outer wall of the city, the nuclear envelope is the reinforced vault of the central bank. Yet, a vault that is completely sealed would be useless. It must be able to receive requests and send out instructions. To facilitate this, the nuclear envelope is perforated by thousands of tiny openings called nuclear pores. These pores are far more complex than simple holes. Each one is ringed by a massive protein complex that acts like a sophisticated security detail. These proteins decide with absolute precision which molecules are allowed to enter and which are allowed to exit. Small molecules might slip through easily, but large proteins or strands of genetic information require a specific chemical passport to pass. This ensures that the delicate machinery of the nucleus is never disturbed by the wrong visitors. Once we pass through these pores, we enter the nucleoplasm, the specialized fluid that fills the nucleus. Suspended within this fluid is the reason for the nucleus's existence, the DNA deoxyribonucleic acid. DNA is the master blueprint of life, containing every instruction necessary to build and operate you. If you were to take the DNA from a single human nucleus and stretch it out into a straight line, it would be about 2 meters long. This creates a fascinating mechanical problem. How does the cell fit 2 meters of string into a space that is only about 6 micrometers wide? The answer lies in an extraordinary feat of biological packaging. The DNA is not simply shoved into the nucleus like a tangled ball of yarn. Instead, it is carefully wrapped around specialized proteins called histones, much like thread is wrapped around a spool. This complex of DNA and protein is called chromatin. Most of the time, as you lie here resting, your chromatin exists in a loose, diffused state, looking like a blurry, thread-like mass under a microscope. This looseness is intentional. It allows the cell's machinery to access the text of the DNA so it can be read and copied. However, when it comes time for a cell to divide and create a new life, the chromatin coils up even tighter, condensing into the thick, visible structures we know as chromosomes. This transformation is like packing the library's scrolls into sturdy crates before moving them to a new building, ensuring that not a single word of the instructions is lost or broken during the move. If we look even closer into the nucleoplasm, we often see a dark, dense circular region that stands out from the rest of the chromatin. This is the nucleolus. It is a fascinating structure because, unlike most organelles, it is not surrounded by its own membrane. It is simply a specialized region where the cell's most urgent manufacturing takes place. The nucleolus is a factory dedicated to the production of ribosomal RNA, rRNA. This is where the components of ribosomes, the protein-building machines we will meet in our next lesson, are constructed. You might think of the nucleolus as a specialized workshop within the architect's office. The architect, the DNA, provides the plans, and the workshop, the nucleolus, builds the workers, the ribosomes, that will eventually carry those plans out into the rest of the cell. Once the parts of the ribosomes are assembled in the nucleolus, they pass through the nuclear pores and out into the cytoplasm, where they begin their lifelong task of building proteins. Take a moment to let that idea find its place. The nucleus is not just a storage site. It is a center of constant, quiet activity. It is the site of a process called transcription. Because the original DNA blueprints are too precious to ever leave the vault, the nucleus makes a working copy of the instructions. This copy is a single-stranded molecule called messenger RNA, or mRNA. Think of this like a student who goes to a rare books library. They aren't allowed to check out the ancient handwritten manuscript, but they are allowed to use a photocopier to take a page home with them. That photocopy is the mRNA. It carries the genetic message out through the nuclear pores and into the cytoplasm, where the ribosomes can read it. This ensures the original DNA remains safe and pristine inside the nuclear envelope, protected from the chemical reactions and potential dangers of the wider cell. The discovery of the nucleus marks a turning point in the history of science. It was first described in detail by the Scottish botanist Robert Brown in 1831 while he was studying orchids. At the time, Brown didn't fully understand what the dark spot did, but he recognized it was a constant feature of almost every plant cell he examined. It took nearly another century for us to realize that this spot held the secret to heredity. We now know that every eukaryotic cell, with a few exceptions like your mature red blood cells, contains this central archive. And because the nucleus is so well protected, it is the part of the cell that allows us to look back in time. When scientists study ancient remains, it is the DNA protected within the nuclei of bone and tooth cells that allows us to map the history of our species. So, why is the nucleus so central to your life as you sleep tonight? This isn't just a theory of structures. It is the principle that explains why you are uniquely you. The DNA inside your nuclei contains the code for your eye color, the shape of your hands, and even the way your body responds to the rhythm of the night. The nucleus is also the front line in our understanding of aging and disease. As we grow older, the DNA inside our nuclei can accumulate tiny nicks or errors from exposure to the environment. While the nucleus has a remarkable set of repair enzymes that act like librarians fixing torn pages, sometimes errors slip through. This is the root of many cancers, when the genetic instructions for growth become corrupted, and the nucleus begins telling the cell to divide uncontrollably. By studying the nucleus, we are learning how to catch these typos in the genetic code and fix them before they cause harm. Consider also the miraculous fact of cellular differentiation. As I mentioned earlier, almost every cell in your body contains the exact same set of DNA. The nucleus in your skin cell has the instructions on how to make a heart, and the nucleus in your heart cell has the instructions on how to make skin. The reason they are different is because of the nucleus's role as a controller. It doesn't read the whole book at once. In a skin cell, the nucleus keeps the heart chapters tightly coiled and inaccessible, while keeping the skin chapters open and ready to be copied into mRNA. It is the world's most sophisticated editing process, allowing a single set of blueprints to build 1,000 different types of rooms. If your mind is still following along, try visualizing this. Inside you, trillions of these tiny dark spheres are quietly managing the library of your life. They are ensuring that your heart keeps its rhythm and your lungs keep their breath, all by carefully reading the right page at the right time. Don't worry about the complexity. Just let the logic of this central control wash over you. You are being guided from within by a system of incredible wisdom and order. Having now grasped the structure of the central vault and the way the blueprints are protected and copied, we are perfectly prepared to appreciate what happens next. The nucleus has made its photocopies, the mRNA, and sent them out through the nuclear pores. But where do these instructions go? Who is waiting on the other side of the vault door to receive them? Let's hold that thought here, a complete piece of the puzzle. We have explored the brain of the cell. Our next lesson will hand us the adjacent piece, which is the study of ribosomes and protein synthesis. We will leave the quiet halls of the nucleus and follow the mRNA into the bustling workspace of the cytoplasm. There, we will meet the tiny, tireless machines that read the genetic code and turn it into the physical proteins that build your body and power your dreams. The blueprints have been issued. The work is about to begin. For now, rest in the knowledge that the center is holding. Your genetic legacy is safe, and the instructions for your well-being are being carried out with perfect precision. Lesson 5. Ribosomes and Protein Synthesis. We have spent our recent time together within the hushed, protected corridors of the nucleus, marveling at the ancient archives of DNA. We have seen how the master blueprints are kept under the double membrane lock of the nuclear envelope. But as any architect will tell you, a blueprint is merely a set of possibilities until it is handed to a builder. Tonight, we move from the archive to the assembly line. We follow the messenger, RNA, that fragile, silver-threaded copy of our genetic intent, as it slips through the nuclear pores and enters the vast, industrious space of the cytoplasm. In this lesson, we will meet the builders of the cellular world, the ribosomes. These are perhaps the most humble-looking yet profoundly important structures in all of biology. They are the sites of protein synthesis, the miraculous event where the language of nucleic acids is translated into the physical language of proteins. Without these tiny, tireless machines, our genetic code would be a silent book that no one could read. We will explore how these workers are built, how they read the genetic tape, and how they stitch together the long chains of amino acids that eventually become your muscles, your enzymes, and your very thoughts. To understand the ribosome, we must first look at its nature. Unlike the nucleus or the mitochondria, a ribosome is not an organelle enclosed in its own membrane. Instead, it is a complex molecular machine, a dense aggregate of ribosomal RNA and various proteins. You might think of it not as a room in our cellular mansion, but as a specialized piece of equipment, perhaps a high-precision 3D printer, that can be moved to wherever the work is most needed. Every ribosome consists of two distinct parts, the large subunit and the small subunit. When they are not working, these two halves often float separately in the cytoplasm, like two parts of a clasp waiting to be joined. They only come together when a strand of messenger RNA, mRNA, arrives from the nucleus. At that moment, the small subunit latches onto the mRNA tape, and the large subunit snaps into place on top of it. The process that follows is known as translation. It is one of the most elegant transitions in the natural world. The mRNA carries the genetic code in a series of three-letter words called codons. Each codon is a specific instruction for a single amino acid. However, when the ribosome cannot simply reach out and grab these amino acids, it needs a translator. This role is played by another type of molecule called transfer RNA, or tRNA. Imagine the tRNA as a specialized courier. On one end, it carries a specific amino acid, the raw building material. On the other end, it has a three-letter anticodon that acts like a key. The ribosome acts as the workbench where the mRNA tape is pulled through. As each codon on the tape is exposed, the ribosome waits for the tRNA with the matching key to arrive. When the correct tRNA lands, the ribosome takes the amino acid it is carrying and snips it off, attaching it to a growing chain of amino acids. This chain is called a polypeptide. As the ribosome moves along the mRNA, one step at a time, the chain grows longer and longer, and it is like a master weaver pulling a thread through a loom, adding a single bead at a time according to a pattern. Once the ribosome reaches a stop codon, a specific signal that the instruction is complete, it releases the polypeptide chain, and the two subunits of the ribosome drift apart, ready to start the process again elsewhere. Take a moment to let that image find its place in your mind. Trillions of these tiny looms are at work within you right now, translating the abstract instructions of your DNA into the physical reality of your body. They are the bridge between the idea of you and the fact of you. While all ribosomes perform the same basic task of protein synthesis, they are not all found in the same place. In the eukaryotic cell, we find them in two primary locations, and their location tells us a great deal about the destiny of the proteins they are building. The first group consists of free ribosomes. These are suspended in the cytosol, the jelly-like fluid of the cell. Generally, these ribosomes make proteins that will function right there in the fluid. These might be enzymes that help break down sugars for energy, or proteins that maintain the internal scaffolding of the cytoskeleton. You might think of these as the local contractors who build things for the interior of the house. The second group are bound ribosomes. These are attached to the outside of a sprawling membrane system called the endoplasmic reticulum. When a ribosome is bound to this membrane, it injects its growing polypeptide chain directly into the interior of the reticulum. These proteins are usually destined for more global roles. They might be packaged into vesicles to be sent out of the cell entirely, like the insulin made in your pancreas. Or they might be inserted into the cell's own plasma membrane to act as receptors or gates. This brings us to a beautiful historical realization. For many years, we didn't understand how a ribosome knew whether to stay free or bind to the membrane. In the 1970s, a scientist named Günter Blobel discovered that the first few amino acids of a growing protein chain act as a zip code or a signal sequence. If the ribosome starts building a protein with a specific mailing address on its tip, a special particle recognizes it and drags the whole ribosome over to the endoplasmic reticulum to finish the job. For this discovery, Blobel was awarded the Nobel Prize, reminding us that even at the microscopic level, life is governed by a highly organized postal system. It is also important to recognize that ribosomes are incredibly fast. A single ribosome can add about 2 amino acids to a chain every second. Furthermore, a single strand of mRNA is rarely read by just one ribosome at a time. Instead, many ribosomes will latch onto the same mRNA tape, one after the other, forming what we call a polyribosome. It looks like a string of pearls, with each pearl building its own copy of the same protein. This allows the cell to churn out a high volume of a specific protein very quickly in response to a signal from the nucleus. Think of the scale of this. In a single human cell, there may be several million ribosomes. They are the most numerous machines in your body. They are the reason you can heal a cut, and grow your hair, or produce the antibodies that fight off a late-night chill. They are the universal workers, found in every form of life, from the simplest bacterium to the most complex human. So, why should we care about these tiny dots of protein and RNA as we drift towards sleep? Because the ribosome is the ultimate target for much of our modern medicine. Many of the most effective antibiotics, such as tetracycline or erythromycin, work by specifically attacking the ribosomes of bacteria. Because bacterial prokaryotic ribosomes are slightly smaller and differently shaped than your own eukaryotic ribosomes, these medicines can jam the bacterial machinery without affecting yours. The antibiotic acts like a wrench thrown into the gears of the bacterial factory. If the bacteria cannot make proteins, they cannot build cell walls. They cannot move, and they cannot reproduce. Your immune system can then easily clear them away. Furthermore, we see the importance of ribosomes in the world of biotechnology. When we produce life-saving drugs, like synthetic insulin or growth hormones, we are essentially hijacking the power of the ribosome. We take the human gene for insulin, insert it into a bacterium or a yeast cell, and let that organism's ribosomes do the hard work of building the protein for us. We have turned the ribosome into a microscopic manufacturer that works for the benefit of human health. As you lie here, your own ribosomes are engaged in a vital nighttime shift. During sleep, your body enters a state of repair and growth. Growth hormone levels rise, signaling the nucleus to send out more mRNA instructions to the cytoplasm. Your ribosomes respond by accelerating their work, repairing damaged muscle fibers, building new skin cells, and replenishing the enzymes your brain used during the day. If your mind is still following along, try to visualize this. A trillion tiny weavers moving with rhythmic precision, following the ancient silver threads of your DNA to keep the mansion of your body in good repair. Don't worry about the chemistry or the complex names of the amino acids. Just let the logic of this constant, quiet construction wash over you. You are being rebuilt, molecule by molecule, every second of the night. We have seen the builders at work. We have watched the ribosomes translate the code and stitch together the long chains of amino acids. But a long chain of amino acids is like a raw piece of lumber or a bolt of uncut fabric. It is the raw material of a protein, but it is not yet a finished product. It needs to be folded, modified, polished, and shipped to the correct room in the mansion. Let's hold that thought here, a complete piece of the puzzle. We have explored the factory floor. Our next lesson will hand us the adjacent piece, which is the study of the endomembrane system. We will follow the newly made proteins from the ribosomes into the maze of the endoplasmic reticulum and through the shipping docks of the Golgi apparatus. We will see how the cell's industrial complex ensures that every protein reaches its final destination in perfect working order. The raw materials are ready. The assembly line is moving. For now, rest in the knowledge that your body is a master of logistics. Every part of you is being cared for, and every order from the nucleus is being filled with care. Lesson 6. The Endomembrane System. We have observed the tireless builders of our cellular world, the ribosomes, as they translate the silver threads of genetic code into raw chains of amino acids. But as we have noted, a chain of amino acids is merely the raw material of life. It is like a stack of uncut timber at a construction site or a bolt of raw silk in a tailor's shop. To become a functional tool, an enzyme, a hormone, or a structural fiber, that chain must be folded into a precise shape, modified with specialized chemical tags, and delivered to its exact destination. Tonight, we step into the most expansive and industrious part of the cellular mansion, the endomembrane system. This is a sprawling interconnected network of internal membranes that work together in a seamless industrial flow. In this lesson, we will follow the journey of a protein from its birth on the surface of the endoplasmic reticulum through the sophisticated processing centers of the Golgi apparatus, and finally to its destination, whether that be a recycling center or the world outside the cell. We will explore how the cell manages its massive logistics network with a precision that would put any modern shipping company to shame. As you rest, Try to visualize this system as a flowing river of silver membranes, carrying the very substances that maintain your life. The endomembrane system is not a single organelle, but a collection of membranes that are either physically connected or exchange pieces of themselves through tiny traveling bubbles called vesicles. This system includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, all various types of vacuoles, and the plasma membrane itself. Together, they form a unified manufacturing and distribution complex. Our journey begins with the endoplasmic reticulum, or the ER. The word endoplasmic means within the cytoplasm, and reticulum is the Latin word for a little net. It is indeed a vast net, a maze of folded membranous sacs and tubes that account for more than half of the total membrane in many eukaryotic cells. The ER is so extensive that it is often continuous with the outer layer of the nuclear envelope, creating a direct pipeline from the control center to the factory floor. The ER is divided into two distinct regions that serve very different purposes. The first is the rough ER. It earns its name from the thousands of ribosomes that stud its outer surface, giving it a bumpy, its sandpaper-like appearance under a microscope. As we learned in our last lesson, these bound ribosomes are the ones building proteins destined for export or for the cell's own membranes. As the ribosome builds the protein chain, it threads it through a pore into the interior space of the rough ER, which we call the lumen. Inside the quiet sanctuary of the lumen, the protein begins to fold into its functional three-dimensional shape. It may also have short chains of sugars attached to it, turning it into a glycoprotein. Once the protein is ready, the ER membrane pinches off a tiny piece of itself, wrapping the protein in a protective bubble called a transport vesicle. The second region is the smooth ER. It lacks ribosomes, giving it a sleek, polished appearance. If the rough ER is a textile mill, the smooth ER is a chemical laboratory. Its functions are diverse and essential. It is the site where the cell synthesizes lipids, including the phospholipids that make up all our membranes, and the steroid hormones that coordinate our body's growth and stress responses. In your liver cells, the smooth ER is also the primary site of detoxification, using specialized enzymes to break down drugs and poisons. Furthermore, the smooth ER acts as a warehouse for calcium ions, which are released in a sudden burst whenever a muscle cell needs to contract. Once a transport vesicle buds off from the rough ER, it drifts through the cytosol, often guided by the microtubule tracks we discussed, until it reaches the next major station, the Golgi apparatus. If the ER is the factory, the Golgi is the warehouse, the finishing shop, and the shipping department all in one. It consists of a stack of flattened membranous sacs that look somewhat like a pile of pita bread. These sacs are not physically connected like the ER. Instead, they exist as a series of distinct stages. The side of the Golgi facing the ER is called the sysface, which acts as the receiving dock. The vesicle from the ER fuses with this membrane, emptying its contents into the Golgi's interior. As the proteins move through the stacks of the Golgi, they undergo further refinement. Enzymes within the Golgi might trim some sugar chains or add others, acting like a craftsman putting the final polish on a piece of furniture. Most importantly, the Golgi serves as a sorting center. It attaches molecular shipping labels, often small phosphate groups, to the proteins, ensuring the cell knows exactly where they belong. Finally, on the transface, or the shipping dock, the proteins are repackaged into new vesicles and sent to their final destination. While many proteins are sent out to the plasma membrane for secretion, others are destined for internal roles. One of the most important products of the Golgi is the lysosome. A lysosome is a membrane-enclosed sac filled with powerful hydrolytic enzymes designed to break things down. You can think of the lysosome as the cell's stomach and its recycling center. These enzymes work best in an acidic environment, so the lysosome maintains an internal pH that is much lower than the rest of the cell. This is a brilliant safety feature. If a lysosome were to accidentally break open, its enzymes would be less active in the neutral pH of the cytosol, preventing them from digesting the cell itself. Lysosomes perform two vital roles. The first is phagocytosis, or cellular eating. When a cell engulfs a piece of food or a stray bacterium, it forms a food vacuole. A lysosome then fuses with this vacuole, releasing its enzymes to break the contents down into simple sugars and amino acids that the cell can use for fuel. The second role is autophagy, which literally means self-eating. The lysosome seeks out damaged organelles, like a mitochondrion that has stopped working, wraps them in a membrane, and breaks them down. This allows the cell to constantly renew itself, recycling its own organic material to build new structures. In plant cells and fungi, we see a variation of this system called the vacuole, while animal cells have small, temporary vacuoles. A mature plant cell often contains one massive central vacuole that can take up 80% or more of the cell's volume. This vacuole is a versatile storage tank. It holds water to maintain the plant's structural turgor pressure, the reason a watered plant stands upright, stores ions, and can even sequester poisonous compounds that protect the plant from being eaten by herbivores. The entire endomembrane system is a study in the dynamic nature of life. We often think of organelles as permanent structures, but in this system, the membranes are constantly flowing and changing. A piece of membrane that was part of the rough ER 10 minutes ago might now be part of a transport vesicle. 10 minutes from now, it might fuse with the Golgi, and eventually, it may become part of the plasma membrane itself. Historically, we owe much of our understanding of this system to Camillo Golgi, an Italian physician who, in 1898, discovered the apparatus that bears his name. He used a specialized silver staining technique that allowed him to see this internal reticular apparatus for the first time. For decades, many scientists thought what he saw was an illusion created by the stain, but the electron microscope eventually proved him right. His discovery opened our eyes to the fact that the cell is not just a bag of parts, but a coordinated assembly line of incredible sophistication. If your mind is still following along, try to visualize the sheer scale of this logistics network as you lie in the quiet of the night. This system is the reason you can maintain your health and interact with your world. Consider the cells in your stomach lining. They are packed with rough ER and Golgi stacks because their entire life is dedicated to the endomembrane system. They are constantly synthesizing digestive enzymes and packaging them into vesicles and shipping them to the surface to be released so you can digest your dinner. Without the endomembrane system, these enzymes would be trapped inside the cell, where they would be useless or even harmful. We also see the importance of this system in immune health. Your B cells, the white blood cells that produce antibodies, are masters of the endomembrane system. When you are fighting an infection, a single B cell can produce and secrete up to 2,000 antibody proteins every second. To do this, the cell must drastically expand its rough ER and Golgi apparatus, turning itself into a high-speed factory dedicated to the shipping of these protective proteins. When this system fails, the consequences are profound. There is a group of conditions known as lysosomal storage diseases, such as Tay-Sachs disease. And in these cases, a person is born without one of the specific enzymes found in the lysosome. Because the recycling center is missing a tool, waste products, specifically certain lipids, begin to build up inside the cells of the brain. Eventually, this accumulation interferes with the cell's function, leading to severe neurological decline. It is a sobering reminder that the quiet work of sorting, packaging, and recycling is what keeps the chaos of the world at bay. As you sleep, your endomembrane system is particularly active. This is the time when your body focuses on autophagy, the internal house cleaning where lysosomes break down the cellular wear and tear of the day. It is a period of restoration and renewal, where the factories of the ER and the shipping docks of the Golgi work together to ensure that by morning, every room in your biological mansion is replenished and ready for the day ahead. We have toured the great industrial complex of the cell. We have followed the flow of membranes from the nucleus to the ER, through the Golgi, and out to the lysosomes and the plasma membrane. We have seen how the cell builds, modifies, and ships the molecules of life. But all of this activity, the constant building of proteins, the movement of vesicles along the cytoskeleton, the pumping of ions into vacuoles, requires an immense and steady supply of energy. A factory cannot run without power, and a city cannot thrive without a grid. Let's hold that thought here, a complete piece of the puzzle. We have seen the manufacturing and shipping departments. Our next lesson will hand us the adjacent piece, which is the study of mitochondria and cellular respiration. We will explore the powerhouse of the cell, where the energy stored in your food is converted into the universal currency that powers every single process we have discussed so far. The assembly lines are moving, and the packages are being delivered. For now, rest in the knowledge that your internal world is a masterpiece of efficiency and grace. You are being renewed from within, molecule by molecule, in the quiet of the night. Lesson 7. Mitochondria and Cellular Respiration. We have traveled through the bustling industrial corridors of the endomembrane system, observing the intricate ways in which the cell builds, folds, and ships the molecules of life. We have seen the ribosomes at their looms and the Golgi apparatus at its shipping docks. But as we have noted, no factory can run without a power source, and no city can survive without a constant flow of electricity. Every movement of a vesicle, every stitch of a protein chain, and every beat of your heart requires an immediate and reliable source of energy. Tonight, we turn our attention to the powerhouse of the cell, the mitochondrion. In this lesson, we will explore this fascinating organelle, which exists as a world within a world. We will look at its unique, double-membraned structure and discover how it acts as a biological furnace, burning the sugars from your food to create the universal energy currency known as ATP. We will also touch upon the mysterious history of the mitochondrion, the evidence that it was once an independent traveler that joined forces with our ancestors billions of years ago. As you settle into the stillness of the night, try to visualize these thousands of tiny glowing embers within every cell of your body, quietly and efficiently fueling the miracle of your existence. To understand how the cell powers itself, we must look at the structure of the mitochondrion. These organelles are found in nearly all eukaryotic cells, from the smallest fungi to the most complex human. While a cell might have only one nucleus, it can have hundreds or even thousands of mitochondria, not depending on its energy needs. A muscle cell, which must be ready to exert force at a moment's notice, is packed with these powerhouses, while a less active cell might have far fewer. The mitochondrion is defined by its two distinct membranes. The outer membrane is smooth and acts as a simple protective skin, but the inner membrane is a masterpiece of biological architecture. It is folded back and forth upon itself in complex convolutions called cristae. This folding is not accidental. It is a brilliant solution to a mathematical problem. By folding the membrane, the mitochondrion creates a massive amount of surface area within a very small space. This is essential because the inner membrane is where the actual machinery of energy production is located. The more surface area there is, the more power the cell can generate. This inner membrane divides the mitochondrion into two internal compartments. The first is the intermembrane space, the narrow gallery between the outer and inner walls. The second, and most important, is the mitochondrial matrix. This is the dense fluid enclosed by the inner membrane. The matrix contains a unique mixture of enzymes, as well as the mitochondrion's own DNA and ribosomes. The primary function of the mitochondrion is a process called cellular respiration. You might think of this as a slow, controlled burning of food. When you eat, your body breaks down carbohydrates into a simple sugar called glucose. This glucose is delivered to your cells, but it is too raw to be used directly as fuel. It is like having a pile of crude oil. It has potential, but you cannot put it directly into a delicate machine. The mitochondrion takes this glucose and, in the presence of oxygen, breaks it down through a series of complex chemical steps. As the chemical bonds in the sugar are broken, energy is released. The mitochondrion captures this energy and uses it to recharge a molecule called ATP. Adenosine triphosphate. ATP is the universal energy currency of life. If a protein pump in the membrane needs to move a sodium ion, it spends an ATP molecule to get the job done. If a motor protein needs to take a step along a microtubule, it spends an ATP. The mitochondrion is the bank where this currency is minted. It takes the low-energy spent version of the molecule, ADP, and adds a phosphate group back onto it, creating a high-energy ATP molecule ready for use. This cycle of spending and recharging happens millions of times a second in every cell of your body, even as you lie perfectly still. If we look closer at the process of cellular respiration, we see a beautiful three-act play. The first act, glycolysis, happens outside the mitochondrion, in the cytosol. It breaks the glucose in half, providing a small amount of energy. The second act, the citric acid cycle, also known as the Krebs cycle, takes place inside the mitochondrial matrix. Here, the remains of the glucose are completely dismantled, releasing carbon dioxide as a waste product, the very carbon dioxide you breathe out into the night air. The final act, the electron transport chain, is the climax of the story. It takes place on the inner membrane, specifically on those folded cristae. This is where the majority of the ATP is produced. Imagine a series of protein complexes embedded in the membrane like a row of water wheels. High-energy electrons, stripped from the food during the previous acts, are passed from one wheel to the next. As the electrons move, they provide the energy to pump hydrogen ions, protons, into the intermembrane space, creating a high-pressure reservoir of ions. Finally, these ions rush back through a specialized protein called ATP synthase, which acts like a microscopic turbine. As the ions flow through, the turbine spins, and that mechanical motion is used to snap a phosphate onto ADP, creating ATP. It is a literal hydroelectric dam operating at the molecular scale. One of the most profound aspects of mitochondria is their history. As I mentioned, they possess their own DNA, small, circular chromosomes that look very much like the DNA of bacteria. They also have their own ribosomes, and they divide on their own schedule, independent of the rest of the cell. This led the scientist Lynn Margulis to champion the endosymbiotic theory in the late 1960s. She proposed that billions of years ago, an ancestral eukaryotic cell, which was probably a large, slow-moving predator, swallowed a smaller, aerobic bacterium. However, instead of being digested, the bacterium survived. It found a safe, nutrient-rich home inside the larger cell, and in return, it provided a massive amount of energy by using oxygen, a skill the larger cell lacked. Over eons, this partnership became so tight that the two could no longer live apart. The bacterium became the mitochondrion. This means that you are, in a very real sense, a chimera, a fusion of different lineages of life. The DNA in your nucleus comes from your parents, but the DNA in your mitochondria is a direct, unbroken line from your mother, your grandmother, and back through a billion years of female ancestors to that original ancient bacterium. Take a moment to let that idea find its place. You carry an ancient traveler within you, fueling your dreams with a legacy older than the mountains. So, how does the work of the mitochondrion manifest in your life as you sleep? This organelle is the bridge between the air you breathe and the life you live. Every breath you take is primarily for the benefit of your mitochondria. Oxygen is the final electron acceptor in the electron transport chain. It sits at the bottom of the water wheel, catching the electrons so the cycle can continue. Without oxygen, and the wheels stop turning, the ATP bank closes, and the cell's activities grind to a halt. This is why you cannot survive more than a few minutes without air. The efficiency of your mitochondria is also the secret to your metabolic health. When you are physically active, your cells demand more ATP. Your mitochondria respond by working harder, and even by dividing to create more powerhouses. This is why endurance athletes have a much higher density of mitochondria in their muscles than sedentary individuals. Their bodies have built a larger power grid. Conversely, we are beginning to understand that many chronic conditions, from type 2 diabetes to certain types of fatigue, are rooted in mitochondrial dysfunction. If the furnaces of the cell begin to leak or become inefficient, they produce harmful byproducts called free radicals, which can damage the cell's DNA and membranes. This oxidative stress is one of the primary drivers of the aging process. By protecting our mitochondria through healthy habits and rest, we are essentially maintaining the power plants that keep our biological city from falling into decay. Consider also the miraculous nature of your heart. It is a muscle that never rests, beating over 100,000 times a day. To do this, heart cells must be among the most mitochondrially dense cells in the body, with these powerhouses taking up nearly 35% of the cell's volume. While you sleep, your heart continues its steady rhythm, powered by a tireless flow of ATP generated by these ancient endosymbionts. If your mind is still following along, try to visualize this. Thousands of these bean-shaped powerhouses in every cell, their inner membranes folded into intricate labyrinths, their molecular turbines spinning with a speed we can barely imagine. They are taking the quiet breaths you draw and turning them into the energy that maintains your body temperature, repairs your tissues, and allows your brain to process the events of the day. Don't worry about the complex chemistry of the citric acid cycle. Just let the logic of this energy flow wash over you. You are a being of light and breath, powered by a billion-year-old partnership. We have seen how the cell generates its power. We have explored the mitochondrial furnace and the hydroelectric dam of the inner membrane. We have seen how the spent currency of the cell is recharged so that the work of the mansion can continue. But as we conclude our study of energy in the animal cell, we must look at the larger cycle of life on our planet. Our mitochondria can only recharge ATP if they have a supply of glucose, a fuel source. But where does that fuel come from? To find the ultimate source of all biological energy, we must step outside the human body and look at the world of plants. Let's hold that thought here, a complete piece of the puzzle. We have seen how energy is released. Our next lesson will hand us the adjacent piece, which is the study of chloroplasts and photosynthesis. We will examine the green organelles that capture the light of the sun and turn it into the very sugars that our mitochondria burn. We will see how the solar panels of the plant world provide the foundation for almost all life on Earth. The furnaces are humming, and the grid is stable. For now, rest in the knowledge that you are a vital part of a grand energetic cycle. You are fueled, you are protected, and you are at peace. Lesson 8. Chloroplasts and Photosynthesis. In our last session, we ventured into the inner furnaces of the cell, exploring how the mitochondria act as the power plants for our biological mansion. We learned that these organelles are the reason we breathe, as they use oxygen to break down the sugars in our food into the energy currency of ATP. But as we rest in the quiet of the night, we must ask a fundamental question. Where does that fuel come from? If the mitochondria are the furnaces, who is the provider of the coal? If they are the engines, who is the producer of the gasoline? Tonight, we step outside the boundaries of the human body and into the vibrant green world of plants and algae. We are going to explore the chloroplast, the organelle responsible for the most important chemical reaction on our planet, photosynthesis. This is the process that captures the raw, radiant energy of the sun and weaves it into the physical bonds of sugar molecules. While you do not have chloroplasts in your own cells, your very existence, every breath you take, and every ounce of energy you spend is a direct gift from these solar generators. We will examine the intricate coin-like architecture of the chloroplast, and see how it mirrors the energy production logic we saw in the mitochondria, completing the grand cycle of life that sustains our world. To understand the chloroplast, we must first look at its home. Chloroplasts are found in the green parts of plants, primarily the leaves, and in eukaryotic algae. A single leaf cell might contain anywhere from 20 to 100 of these bright green ovals. Like the mitochondria, the chloroplast is defined by a double membrane envelope. There is a smooth outer membrane and a smooth inner membrane, separated by a very thin intermembrane space. However, unlike the mitochondria, the inner membrane of the chloroplast does not do the heavy lifting of energy production. Instead, the chloroplast contains a third, even more specialized membrane system located deep within its interior. Imagine a large open hall filled with fluid. This fluid is called the stroma. It contains the chloroplast's DNA, its ribosomes, and many essential enzymes. Suspended within this stroma is a network of interconnected sacs called thylakoids. These sacs look remarkably like stacks of green coins or flattened pancakes. An individual coin is a thylakoid, and a whole stack of them is called a granum. And it is within the membranes of these thylakoids that the miracle of photosynthesis begins. Embedded in these thylakoid membranes is a molecule you have likely heard of, chlorophyll. This is the green pigment that gives plants their color. But chlorophyll is more than just a pigment. It is a solar collector. It is designed to absorb specific wavelengths of light, mostly the blues and the reds, while reflecting the green light back to our eyes. When a photon of sunlight strikes a chlorophyll molecule, it excites an electron, kicking it up to a higher energy level. This is the moment where light energy is transformed into chemical energy. The overall process of photosynthesis can be thought of as the reverse of cellular respiration. In respiration, the cell breaks down sugar and oxygen to produce energy, water, and carbon dioxide. In photosynthesis, the chloroplast uses the energy from sunlight to combine carbon dioxide and water to produce sugar and oxygen. The sugar, usually glucose, becomes the food for the plant and eventually for every animal on Earth, while the oxygen is released as a waste product into the atmosphere. Take a moment to let that logic wash over you. The waste of the plant is the life breath of the animal. The solar energy captured by a leaf in the afternoon becomes the power that allows you to dream at night. It is a perfect closed-loop system of extraordinary elegance. If we look closer at how the chloroplast manages this solar harvest, we see that photosynthesis is divided into two distinct stages. The first stage is called the light reactions, and as the name suggests, it can only happen when the sun is shining. These reactions take place on the thylakoid membranes. When the sunlight hits the chlorophyll, the energized electrons are passed along an electron transport chain, very similar to the one we saw in the mitochondria. As these electrons move, they provide the energy to pump hydrogen ions into the interior of the thylakoid sac, creating a high-pressure reservoir. Just like in the mitochondria, these ions eventually rush out through an ATP synthase turbine, generating ATP. But the chloroplast does something else as well. It uses some of that light energy to split water molecules apart. This is a violent act on a molecular level. It strips the electrons from the water to replace the ones lost by the chlorophyll, leaving behind hydrogen ions and oxygen gas. This is the source of almost all the oxygen in our atmosphere. Without the splitting of water in the thylakoid, the Earth would be a suffocating alien world. The second stage of photosynthesis is called the Calvin cycle, and it takes place in the fluid stroma surrounding the thylakoid stacks. This stage does not require light directly, though it depends on the ATP produced by the first stage. In the Calvin cycle, the chloroplast takes carbon dioxide molecules from the air and fixes them, attaching them to existing carbon chains to build sugars. Think of the light reactions as the power plant that generates electricity, and the Calvin cycle as the factory that uses that electricity to manufacture goods. The goods in this case are stable, energy-rich sugar molecules that can be stored for later or used to build the plant's own body, its roots, its stems, and its fruit. Like the mitochondria, the chloroplast has a fascinating evolutionary history that supports the endosymbiotic theory. Chloroplasts have their own circular DNA, their own ribosomes, and they reproduce by dividing in two, much like bacteria. Scientists believe that billions of years ago, a non-photosynthetic eukaryotic cell engulfed a photosynthetic bacterium, specifically a cyanobacterium. Instead of being eaten, the cyanobacterium became a permanent resident, providing its host with a constant supply of food in exchange for protection and raw materials. This event was the birth of the entire plant kingdom. When you look at a majestic forest or a simple blade of grass, you are looking at the descendants of that ancient successful partnership. You are looking at a world made possible by a single lucky merger between two microscopic strangers. If your mind is still following along, try to visualize the sheer scale of what is happening in the leaves of the trees outside your window. Every year, the chloroplasts on Earth convert about 250 billion tons of carbon dioxide into organic matter. This is the lung of the planet, pulling in the greenhouse gases that would otherwise overheat our world, and breathing out the oxygen that allows complex life to flourish. The work of the chloroplast is the foundation of our entire global economy. Every piece of wood in a house, every thread of cotton in a shirt, and every calorie in a meal began as a photon of light captured by a chloroplast. Even the fossil fuels we burn today, the coal, oil, and gas, are nothing more than buried sunlight, the remains of ancient plants and algae that used their chloroplasts to harvest the sun millions of years ago. We are literally running our modern world on the recycled energy of prehistoric thylakoids. Consider also the color of our world. Why is the Earth green? It is green because the chloroplast's chlorophyll is so efficient at absorbing the blue and red parts of the spectrum that it has no use for the green light, reflecting it away. If our sun emitted a different kind of light, or if life had evolved a different pigment, the forests might be purple or red. The very aesthetics of our planet are a byproduct of cellular physics. As you lie here, your body is a participant in this solar legacy. The glucose being burned by your mitochondria right now was likely assembled by a chloroplast only a few months or years ago. Perhaps it was in a stalk of wheat, in a sun-drenched field, or a leaf of spinach in a garden. The carbon atoms that make up your own muscles were once floating in the air as carbon dioxide before a chloroplast grabbed them and turned them into life. Don't worry about the complex names of the enzymes like Rubisco. Just let the logic of this energy capture wash over you. You are connected to the sun by a chain of green organelles. You are a solar-powered being once removed. The planet breathes in, the planet breathes out, and you are part of that steady, ancient rhythm. We have completed our tour of the energy centers of life. We have seen how the chloroplast captures the sun to build the fuel, and how the mitochondrion burns that fuel to power the cell. We have seen the incredible symmetry of the biological world, a cycle of carbon, oxygen, and energy that links every living thing together. But a collection of organelles, even ones as powerful as these, does not make a complex organism. To build a person, or a tree, or even a simple tissue, cells cannot exist as isolated islands. They must hold on to each other, communicate with each other, and share the space between them. Let's hold that thought here. A complete piece of the puzzle. We have seen the energy department. Our next lesson will hand us the adjacent piece, which is the study of the extracellular matrix and cell junctions. And we will look at what happens just outside the plasma membrane, how cells use protein cables and specialized handshakes to bind themselves into the strong functional tissues that make up your body. The sun has been captured, and the energy is flowing. For now, rest in the knowledge that you are a child of the light, supported by 1,000,000,000,000 silent, green miracles. You are part of the grand design, and all is well. Lesson 9. The extracellular matrix and cell junctions. We have spent much of our journey deep within the interior of the cell, marveling at the libraries of the nucleus, the assembly lines of the ribosomes, and the power plants of the mitochondria. We have explored the living mansion as if it were a solitary structure standing alone in a vast landscape. But as we move towards the conclusion of our study, we must acknowledge a fundamental truth of biology. Life, in its most complex and beautiful forms, is a collaborative effort. A single cell is a marvel, but a human being is a symphony of trillions of cells working in perfect physical and chemical unison. In this lesson, we are going to step outside the front door of the cellular mansion once again to examine the world that exists between cells. We will explore the extracellular matrix, the sophisticated scaffolding of proteins and sugars that acts as the mortar between the biological bricks of your body. We will also examine the specialized cell junctions, the microscopic handshakes, waterproof seals, and communication tunnels that allow individual cells to bind together into the strong functional tissues that make up your skin, your muscles, and your organs. As you lie in the quiet of the night, try to visualize your body not just as a collection of parts, but as a vast interconnected web where every unit is physically tied to its neighbor, creating a whole that is far greater than the sum of its parts. In the traditional view of biology, we often focus so much on the living part of the cell that we ignore the non-living space around it. However, in multicellular organisms like us, the space between cells is far from empty. It is filled with a dense organized network called the extracellular matrix, or the ECM. If cells are the inhabitants of a city, the ECM is the pavement, the bridges, and the structural girders that allow the city to hold its shape. The primary ingredients of this matrix are glycoproteins, proteins with short chains of sugars attached, secreted by the cells themselves. The most famous and abundant of these is collagen. Collagen is a remarkable molecule. It is a fibrous protein that forms incredibly strong, interwoven ropes. In fact, collagen accounts for about 40% of the total protein in your body. It provides the tensile strength that prevents your tissues from tearing when they are pulled or stretched. When you touch your skin and feel its resilience, or when you move your joints and feel the strength of your tendons, you are feeling the work of the collagen in your extracellular matrix. Woven into this forest of collagen fibers is another type of molecule called a proteoglycan. These are small proteins with many long-branching carbohydrate chains. They act like a sponge, trapping water molecules to create a hydrated, jelly-like ground substance. This gel fills the spaces between the collagen fibers, helping the tissue resist compression. Together, the strong collagen ropes and the resilient proteoglycan gel create a material that is both tough and flexible, providing the perfect environment for cells to live and grow. But the ECM is not just a passive packing material. It is physically connected to the cell's interior. Cells use specialized proteins called integrins that span the plasma membrane. On the outside, these integrins bind to the fibers of the matrix. On the inside, they bind to the microfilaments of the cytoskeleton. This creates a continuous physical bridge from the outside world directly into the heart of the cell. This connection is vital because it allows the cell to feel its environment. When the matrix is stretched or compressed, the signal is sent through the integrins to the cytoskeleton, allowing the cell to adjust its behavior accordingly. As cells settle into this matrix, they must also fasten themselves directly to their neighbors to form tissues. In animal cells, there are 3 primary ways this is achieved, each serving a distinct mechanical or functional purpose. The first are tight junctions. Imagine 2 neighboring cells whose plasma membranes are pressed so tightly together that they are literally sewn in place by specialized proteins. This creates a continuous, waterproof seal around the cells. Tight junctions are the reason that the fluid in your stomach doesn't leak out into your abdominal cavity, and why your skin acts as such an effective barrier against the outside world. They ensure that if a molecule wants to pass from one side of a tissue to the other, it must go through the cells, where it can be properly filtered and processed, rather than slipping between them. The second are demosomes, often called anchoring junctions. If tight junctions are like waterproof stitching, demosomes are like sturdy rivets or spot welds. They fasten cells together into strong, resilient sheets. These junctions are anchored deep within the cytoplasm by the sturdy intermediate filaments of the cytoskeleton. You will find desmosomes in tissues that are subject to great mechanical stress, such as your heart muscle and your skin. They ensure that even when your heart beats with great force, the individual cells stay firmly attached to one another, preventing the tissue from tearing apart. The third, and perhaps most fascinating, are gap junctions, or communicating junctions. These are not about physical strength or sealing, but about conversation. A gap junction is a tiny, protein-lined tunnel that connects the cytoplasm of one cell directly to the cytoplasm of its neighbor. These channels are large enough for ions, sugars, and amino acids to pass through freely. They allow a whole group of cells to act as a single, coordinated unit. In your heart, for instance, it is the gap junctions that allow the electrical signal for a contraction to sweep through the muscle instantly, ensuring that every cell squeezes at the exact same moment. While animal cells rely on the extracellular matrix, and these three types of junctions, plant cells have a very different strategy for holding themselves together. Because plants do not have a skeleton made of bone, every individual plant cell must provide its own structural support. They do this by encasing their plasma membrane in a rigid cell wall. The plant cell wall is much thicker and stronger than the animal plasma membrane. It is primarily composed of cellulose, a long-chain carbohydrate that is the most abundant organic compound on Earth. Now you know cellulose as the fiber in your diet or the primary component of the paper in a book. This wall protects the cell, maintains its shape, and prevents the cell from bursting if it takes in too much water. However, because this wall is so thick and rigid, it would normally act as a barrier to communication. To solve this, plant cells are perforated by channels called plasmodesmata. These are similar in function to the gap junctions of animal cells, but they are much more extensive. A plasmodesma is an actual bridge of cytoplasm that passes through a hole in the cell walls, connecting two neighbors. This makes the entire plant a continuum where water, nutrients, and even chemical signals can flow from the roots to the highest leaves without ever having to cross a single plasma membrane. Historically, our understanding of these connections has changed the way we view the human body. For a long time, we thought of the interstitial space between cells as a simple, static fluid. It wasn't until the 1970s and 80s that researchers like Mina Bissell began to prove that the extracellular matrix actually talks to the nucleus. Bissell showed that if you take a mammary gland cell and place it in a petri dish without its matrix, it stops acting like a mammary cell. But if you provide it with the correct ECM proteins, the integrins send a signal to the nucleus. The breast milk genes are turned on, and the cell begins its work. This discovery proved that the non-living space outside the cell is just as important as the DNA inside. The matrix provides the context for life. It tells the cell where it is and what its job should be. If your mind is still following along, try to visualize this. You are a living fabric. Every tissue in your body is a tapestry woven from cellular threads and extracellular ropes. Your bones are a matrix of collagen that has been hardened with calcium minerals. Your cartilage is a matrix of proteoglycans that acts as a shock absorber. Your blood is a matrix that has remained liquid. You are held together by a beautiful, invisible geometry of proteins and sugars. So, why is this extracellular architecture so vital as you sleep? This is the system that maintains the physical integrity of your body. Consider the sensation of aging. As we mentioned in earlier lessons, the wrinkles that appear on our skin as we grow older are primarily a reflection of changes in the extracellular matrix. Over time, and the collagen fibers become less numerous and less organized, and the gel of the proteoglycans loses its ability to hold water. The skin becomes less resilient and begins to sag. Many of the treatments we use in modern dermatology are designed specifically to stimulate the cells, called fibroblasts, to produce new, fresh collagen to rebuild the matrix from the inside out. We also see the vital importance of cell junctions in human health. There are certain autoimmune diseases where the body's immune system mistakenly attacks the proteins that make up the desmosomes in the skin. When these rivets are destroyed, the skin cells can no longer hold on to each other, leading to severe, widespread blistering. It is a powerful reminder that our strength is entirely dependent on these microscopic connections. Furthermore, our understanding of the ECM is at the heart of the new field of regenerative medicine. Scientists are now learning how to build scaffolds that mimic the extracellular matrix. They can take a donor organ, like a heart, and use a gentle detergent to wash away all the living cells, leaving behind only the ghost-like white frame of the collagen matrix. They can then reseed this frame with a patient's own stem cells. The cells feel the collagen through their integrins, recognize the shape of the heart, and begin to rebuild the organ. We are learning to speak the language of the matrix to heal the body. As you sleep, your cells are busy maintaining these connections. This is the time when the fibroblasts in your skin and tendons are most active, repairing the mortar that was stressed during the day. Your tight junctions are holding the line in your digestive tract, and your gap junctions are allowing your heart and lungs to maintain their steady, rhythmic dialogue. Don't worry about the complex names of the glycoproteins. Just let the logic of this interconnectedness wash over you. You are not a collection of separate parts. You are a single, unified being, stitched together with care and reinforced with incredible strength. You are held fast, you are connected, and you are whole. We have explored the physical ties that bind our cells together. We have seen the collagen ropes of the matrix, the waterproof seals of the tight junctions, and the communicating tunnels of the gap junctions. We have seen how the cell is anchored to its world and how it listens to the context provided by its surroundings. But physical connection is only half of the story. For a trillion cells to work together, they must do more than just hold on to each other. They must be able to send and receive complex messages over long distances. They must be able to react to a change in the environment with a single unified response. Let's hold that thought here, a complete piece of the puzzle. We have seen the structure of the community. Our final lesson will hand us the concluding piece, which is the study of cell communication and signaling. We will examine the microscopic language of life, how a chemical whisper at the surface of a cell can trigger a massive change in behavior in the nucleus. We will see how the cell hears, processes, and responds to the world, bringing our entire study of cellular biology to its final harmonious conclusion. The matrix is strong, and the junctions are secure. For now, rest in the knowledge that you are a masterpiece of biological cooperation. You are supported by a trillion silent handshakes. All is well. Lesson 10. Cell communication and signaling. We have traveled through the grand architecture of the cellular mansion, from the protective gates of the membrane to the energetic furnaces of the mitochondria and the vast libraries of the nucleus. We have seen how cells are physically anchored to one another, woven into the strong, resilient fabric of your tissues. But as we arrive at the final chapter of our journey, we must address the most sophisticated mystery of all. How do these trillions of individual units coordinate their actions to become a single, thinking, breathing human being? A city with beautiful buildings and strong roads is still just a collection of stone and steel, unless the people within can talk to one another. In this final lesson, we explore cell communication and signaling. This is the microscopic language of life, a world of chemical whispers and molecular handshakes that allows a cell to sense its environment and respond with perfect precision. We will examine the three-act play of signal transduction, how a message is received at the surface, how it is amplified through a cascade of internal events, and how it ultimately changes the cell's behavior. As you drift toward the deepest part of your rest, try to visualize your body as a vast, shimmering network of information, where every cell is listening, responding, and working in a perfect, silent dialogue to ensure your well-being. In the realm of biology, communication is almost entirely chemical. While we use words and gestures, cells use molecules. These messenger molecules can be proteins, small peptides, amino acids, or even simple gases. The process of a cell sensing and responding to these molecules is called cell signaling, and it follows a remarkably consistent pattern across all forms of life. The story begins with reception. For a signal to be heard, the target cell must have a specific ear for it. This ear is a specialized protein called a receptor, usually located on the plasma membrane. You might imagine the messenger molecule as a key and the receptor as a lock. A cell might be bathed in hundreds of different signals floating through the bloodstream, but it will only respond to the ones for which it has the matching lock. When the signaling molecule, which we call a ligand, binds to the receptor, it doesn't enter the cell. Instead, its touch causes the receptor protein to change its shape. This subtle shift in geometry is the click of the lock turning. It is the moment the message crosses the threshold from the outside world to the internal environment. Once the signal is received, we move into the second act, transduction. This is often the most complex part of the process. Because the messenger molecule stays outside, the information must be passed along a relay team of molecules inside the cell. We call this a signal transduction pathway. Think of this like a game of telephone, but with a crucial difference: it is also an amplifier. One activated receptor might activate 10 secondary messenger molecules inside the cell. Each of those might activate 10 more enzymes, which in turn activate 100 more. By the time the message reaches its destination, a single chemical whisper at the surface has been amplified into a thunderous command within the cytoplasm. This cascade of events, often involving the adding or removing of phosphate groups to proteins, allows the cell to respond with incredible speed and sensitivity. Finally, we arrive at the response. This is the climax of the story, where the signal finally triggers a specific cellular activity. The beauty of cell signaling is the sheer variety of possible responses. The signal might tell the cytoskeleton to rearrange so the cell can move. It might tell an enzyme to start breaking down stored sugar for a burst of energy. Or it might travel all the way into the nucleus, telling the DNA to turn on a specific gene and start building a new protein. Take a moment to let that logic find its place. From a single touch at the membrane, to a falling row of molecular dominoes, to a change in the very heart of the cell. And this is how your body coordinates its growth, its defense, and its daily rhythms. If we look closer at the types of signaling, we see that cells talk over different distances, much like we use different tools for different conversations. The most intimate form is paracrine signaling. This is a local conversation where a cell releases messenger molecules that only travel a short distance to its immediate neighbors. You might think of this as a whisper across a dinner table. A specialized version of this is synaptic signaling, which happens in your nervous system. A nerve cell releases neurotransmitter molecules into a tiny gap, where they are immediately caught by the receptors of the next nerve cell. This allows for the lightning-fast communication that lets you pull your hand away from a hot stove or process a thought. For long-distance communication, the body uses endocrine signaling. Here, specialized cells secrete hormones into the bloodstream. These hormones are like messages in bottles, cast into a vast ocean. They travel through the entire body, but they are only opened by the specific cells that have the correct receptors. This is how the adrenaline produced in your adrenal glands can tell your heart to beat faster and your lungs to breathe deeper, all at the same moment. One of the most fascinating discoveries in the history of cell biology was the identification of G-protein-coupled receptors, or GPCRs. These are the most common type of receptors in your body. They wind back and forth through the plasma membrane 7 times, like a thread sewn through cloth. When a signal binds to them, they activate a G protein on the inside of the membrane, which acts as an on-off switch for the rest of the signaling pathway. In the 1970s and 80s, researchers Robert Lefkowitz and Brian Kobilka worked tirelessly to map these structures. They realized that GPCRs are the primary way we see, smell, and taste. They are also the way we respond to adrenaline and many other hormones. For their work, they were awarded the Nobel Prize in Chemistry in 2012. Their discovery was profound because it revealed that about half of all modern medicinal drugs work by targeting these specific receptors. Every time you take a heart medication, an antihistamine, or even a simple caffeine boost, you are interacting with the GPCR signaling pathways that Lefkowitz and Kobilka helped us understand. If your mind is still following along, try to visualize the incredible precision of these pathways. A cell doesn't just receive one signal at a time, it is bombarded by thousands. It must integrate all of this information, deciding whether to grow, divide, or stay quiet based on the sum of all the whispers it hears. It is a microscopic form of intelligence, an ongoing calculation of life that happens in the dark, fluid spaces of your tissues. So, why is this microscopic language so important to you as you rest tonight? Cell signaling is the ultimate conductor of the symphony of your body. Consider the process of falling asleep. As the light fades, a small gland in your brain, the pineal gland, begins to secrete a hormone called melatonin. This is an endocrine signal. As it travels through your bloodstream, it binds to receptors on cells throughout your brain and body. The signal is transduced, telling your neurons to slow their firing, your heart to lower its pace, and your core temperature to drop slightly. You don't choose to feel sleepy. You are responding to a coordinated chemical signal that is being heard and acted upon by trillions of listeners simultaneously. We also see the vital importance of signaling when the system breaks down. Cancer, at its most fundamental level, is often a disease of cell signaling. A mutation might cause a growth factor receptor to be stuck in the on position, even when no signal is present. The cell hears a constant command to divide, and because the signaling pathway is broken, it cannot stop. Many of our most advanced targeted cancer therapies work by creating molecules that can sit in these broken receptors and turn the signal off, and restoring order to the cellular conversation. Furthermore, consider the miracle of insulin. If you have just eaten, your blood sugar rises. Your pancreas senses this and releases insulin. This signal binds to receptors on your muscle and liver cells, triggering a transduction pathway that moves glucose transport proteins to the cell surface. The cells then open their doors and pull the sugar out of the blood. In type 2 diabetes, this signaling pathway becomes deaf. The receptors are there, but the telephone line of transduction is damaged. By studying these pathways, we are learning how to repair the communication lines and restore health. As you sleep, a different set of signals takes over. Your cells are receiving maintenance signals that trigger the repair of DNA and the synthesis of new proteins. Your immune cells are talking to one another through small proteins called cytokines, coordinating their patrol of your body to clear out any intruders they might find. This is a time of intense quiet communication. Don't worry about the complex names like cyclic AMP or phosphorylation cascades. Just let the logic of this vast network wash over you. You are a miracle of coordination. You are a trillion voices speaking in a language of perfect harmony, all directed toward the single goal of keeping you alive, healthy, and at peace. We have come to the end of our journey through the cell. We have seen the mansion, its walls, its factories, its archives, its power plants, and finally, its language. We have explored the living unit of life from every angle, and I hope you now see yourself with a new sense of wonder. You are not just a person, you are a cosmos. Within you, trillions of lives are being lived every second. Trillions of ribosomes are weaving, trillions of mitochondria are burning, and trillions of receptors are listening. They do not work for themselves. They work for the you that is the sum of them all. As we conclude this crash course, let the details of the organelles and the chemical pathways fade into the background. What remains is the logic and the beauty of life's organization. You are well built, you are well powered, and you are constantly being cared for by the very cells that make you. The lessons are complete. The knowledge has found its place. For now, there is nothing left to learn and nothing left to do. Let your breath become slow and steady. Trust in the silent wisdom of your trillions of cells to carry you through the night. Sleep well in the quiet, coordinated embrace of your cellular world.